Hello, my name is Graham Gardner, and over the next 5 lectures I'm going to be telling you a whole stack about carbohydrate metabolism. There are a number of interrelated pathways that are quite crucial for the metabolism of carbohydrate, and many of them were introduced last year in cell biology. So I'll be going over a short recap of these pathways. But then, very importantly, I'll be delving in more heavily into how they're regulated, because that's really what's quite crucial. In particular, this regulation describes how you're able to respond during starvation, stress, and exercise, and also how you flick over into a storage mode when you're in a well-fed state. So by the end of this series of lectures, we will have covered all of the pathways that you can see here on this screen. All of these will be going into in a fair bit of depth, and also we'll be covering, most importantly, how they're regulated. But to begin with, I want to start by describing some of the basic principles of carbohydrates, their structure and their properties. Carbohydrates are quite crucial, acting as essential building blocks for lipids, RNA and DNA, non-essential amino acids, and also components of glycoproteins and glycolipids. But on top of that, they're also crucial for energy production. Through their oxidation, they generate ATP. In their most fundamental form, they consist of carbon, hydrogen, and oxygen, and sometimes with a little bit of nitrogen attached, in which case you call them an amino sugar. They also contain structural components, either aldehydes or a ketone, making them an aldose or a ketose carbohydrate. And in addition to that, they contain hydroxide groups. Now at this point, I should also indicate that they can be called a few different names. So carbohydrates are also called saccharides and also called sugars. Now I do promise it wasn't me that came up with the 3 different names for these things, which are all actually the same molecule. But nonetheless, in literature, that's what you'll find. Now, in their simplest form, carbohydrates exist as monosaccharides. Now, of course, mono means one, so in effect, I'm saying that these are single sugar molecules. They consist of a chain of carbon atoms, and that chain length can vary from anywhere from 3 to 8 carbons. And in solution, they form these simple ring structures, which you can see here. So this is the simplest form of sugar. When sugar is stored, it is joined up into chains of sugar molecules. And these can exist as disaccharides, or effectively 2 sugar molecules joined together, oligosaccharides consisting of 3 to 11 sugars, or polysaccharides, and of course poly means lots, so in effect these are lots of sugars all joined up into a big long chain. So I'll now give you a quick look at a few of these storage forms. First of all, here's a disaccharide. So here we've got 2 sugar molecules, the first one and the second one, being glucose and fructose, and they are joined by an alpha glycosidic bond. So in combination, this molecule is called sucrose, which is the same sort of sugar that you get on the table, which we mix up in our coffee and our tea. Now, when we consume sugar of this type, the first thing we have to do is cleave this alpha glycosidic bond that you can see here. And that happens in your small intestine. You release an enzyme called sucrase, and that's specific for this molecule, cleaving this bond, releasing the 2 sugar molecules, which can then be absorbed across the small intestinal wall. Now, at this point, I will just highlight the structure of the glycosidic bond, which you can see here. So this is the alpha form of this bond, in which case the OH group lies below the plane of the saccharide ring. And it makes this, I suppose, this U shape that you can see here. Now this does actually matter because enzymes are specific for particular linkages. So in this case, sucrase is quite specific for cleaving this bond. And it's for this reason that you are able to absorb the sugar that is contained in sucrose. But just to expand on this theme a bit, here's another disaccharide. So in this case, you're looking at lactose. I suspect many of you have heard of lactose. It's the carbohydrate that you find in milk. So this is a crucial energy source for suckling animals. But in this case, have a look at the glycosidic bond joining these two sugar molecules together. You can see that it doesn't have that same U shape as the alpha glycosidic bond for sucrose. In this case, it's got like a zigzag structure, which is a beta glycosidic bond. So effectively the anomeric hydroxide group lies above the plane of the sucrose ring. Sucrase and simple acid hydrolysis can't cleave this bond. So can you guess the name of the enzyme that does cleave the bond for lactose, enabling us to absorb the sugar? Yep, it sounds like the name of its substrate. The enzyme is called lactase. So lactase cleaves this bond, enabling you to absorb that sugar. And it makes good sense that lactase is expressed to a fairly high level in young suckling animals. As you get older, your expression of lactase diminishes. And if this falls away too much, then effectively you can't digest and absorb lactose. So if you drink it, it'll effectively go straight through you and come out the end in a big rush. And this is the very basis for lactose intolerance in adults. As they've got older, they diminish in their expression of lactase, and so they're able to digest and absorb lactose less well. Now, I quite like this story because it really highlights just how specific enzymes can be for particular target bonds. So it's a really good one to remember. Lastly, I'd like to describe the polysaccharides. As I said earlier, poly means lots, so these are lots of sugar molecules joined up in big long chains. A great example is amylose and amylopectin. So amylose consists of sugar molecules joined through those glycosidic or alpha glycosidic 1,4 linkages. And amylopectin is a very simple or a very similar molecule, but it's got branch points, what we call alpha 1,6 linkage branch points. And when you combine amylose and amylopectin together, you get what we've all heard of as being starch. And starch is the main way that plants store carbohydrate for their own energy use. Alternatively, in animals, we store carbohydrate as glycogen. You've probably all heard of marathon runners glycogen loading before a big race. They're effectively trying to store lots of carbohydrate in their muscles before exercise. And glycogen consists of lots of glucose molecules, all joined up by these alpha 1,4 linkages. And also similar to starch, it's got alpha 1,6 branch points, making the molecule more complex. And the last polysaccharide in my list that I want to tell you about is cellulose. Now you find lots and lots of cellulose in plants. It's how they make their cell walls. And in this way they quite differ from animals. We actually make our cell walls out of fat, or more specifically a phospholipid bilayer. Plants make them out of cellulose. So when you're looking around at a dry field full of old dried pasture, you're actually looking at dried out cellulose. Now, have you ever considered, given how much sugar is in cellulose, why it is that we can't go around eating dried out hay to get our carbohydrate requirements? Well, it all comes down to how this molecule is joined up. So the carbohydrates, or the sugars, are joined together by beta 1,4 linkages rather than alpha 1,4 linkages. To cleave these bonds, we would need to express an enzyme called cellulase. And put simply, we don't. So the next question is, how do sheep and cattle and other ruminant animals get away with eating dried out dead grass? Well, it's not because they express cellulase in their gut, I can assure you of that. It's actually the microbes that are in their rumen that are actually expressing the cellulase and digesting and breaking down the carbohydrate that's available. Now these microbes extract some of the energy from that carbohydrate for themselves, but they also release volatile fatty acids, in particular acetate, butyrate and propionate, which the ruminant animal can then absorb and themselves use for energy. So this is a really good example of a symbiosis between microbes and the ruminant animal that enables it to get access to a huge supply of carbohydrate. And just to finish with, here's a diagrammatic example of a polysaccharide. In this case, you can see the long chains of sugar molecules all joined up together via these alpha 1,4 linkages. There's also branch points, which you can see here, which are of course those alpha 1,6 linkages. So this is exactly what amylopectin looks like in starch. It's also what glycogen looks like within our muscles. Alternatively, if this was cellulose, you wouldn't be looking at these alpha 1,4 linkages, you'd be looking at beta glycosidic bonds instead. Okay, so that's enough recapping about the basics of carbohydrate. And if you just want to go back over that material and ask yourself a few questions to test whether or not you've understood it, here's some simple ones that you can go over. Okay, so let's now return to our big picture of carbohydrate metabolism. We've covered off on this bit up in the top corner, the carbohydrate structure and properties. And now we need to move on and focus on this bit here, which deals with both the glucose dependence of tissues, but also how we actually supply glucose to those tissues via facilitated passive diffusion, facilitated by our GLUT transporters. Okay, so let's start by looking at the glucose dependency of tissues and how we maintain glucose homeostasis. Now, glucose concentration in your blood is really tightly controlled. It's held within a very limited homeostatic range. So for monogastrics like us, that's humans, it's maintained within a range of about 4 to 5 millimolars. And for ruminants, such as sheep, it's maintained down in a range of about 3 to 4 millimolars. When these plasma glucose concentrations become too low, we call it hypoglycemia. And these are when the plasma glucose levels get down to less than half normal values. And this can be quite dangerous because glucose-dependent tissues, like your brain, really start to struggle if they are deprived of glucose at these low levels. And in monogastrics, a value of 2 millimolars is pretty bad. And if you get down to 1 millimolar or lower, you're in real strife. And classically, people start to pass out at levels of that concentration. Now, the opposite of hypoglycemia is hyperglycemia. And that's when glucose concentrations in your blood get too high. And this is classically defined as concentrations exceeding 10 millimolars in your blood for an extended period of time. If this happens, it can lead to all sorts of complications. When glucose gets in high concentrations in solution, it starts to precipitate onto things. So, for example, you get glycosylation of a whole range of proteins within your body. A classic one are the crystallins on the lens of your eyes, making you go blind. You can also get glycosylation within the small capillaries. And of course, the biggest impact of this is in your extremities. And this can lead to diminished blood flow into those tissues. Now, all of these are the longer-term outcomes of type 2 diabetes. And I'll bet that everyone here has heard of that particular disease, as it gets a lot of media attention these days. Now, diabetes itself is a fascinating story because it comes in two forms. The first is type 1 diabetes, otherwise known as juvenile onset diabetes, because it manifests pretty early on in life. Now, in this instance, you're usually secreting not enough insulin or dysfunctional insulin. And therefore, this can be regulated by simply injecting the stuff. But the other form of diabetes is called mature age diabetes or type 2 diabetes. Now, this is caused by a loss of insulin sensitivity. It manifests itself in older people, often whom are carrying too much weight, but also who have exposed themselves to too much sugar intake throughout their lives. Big spikes of sugar are followed by corresponding big spikes in insulin, and over time this desensitizes your body to that hormone, and you gradually become less responsive to it. So as a result, you end up not being able to store glucose from your blood, and therefore it stays in solution in your blood at elevated concentrations, or hyperglycemia. And if left unchecked over an extended period of time, this can be quite lethal. And this disease is becoming more and more commonplace in affluent Western societies, classically all of whom are consuming too much sugar on a day-to-day basis. I can actually illustrate the effect of type 2 diabetes using the following graph. What you're looking at here is an oral glucose tolerance test. And believe it or not, this was actually done in our biochemistry labs. We used to run these labs for the biomedical science students. So we would pick a number of participants, tell them to go away and fast overnight on the night before they undertook the laboratory class the next day. When they entered the lab, they would undertake their very first blood glucose reading, which you can see here at time 0. They would then consume a single bolus of 75 grams of glucose, and we could then monitor their blood glucose concentration over the following 2 hours. And you get this nice peak in glucose concentration, and then it starts to decline again as you approach the 120-minute mark. But have a look at these guys up here. So these individuals consumed that glucose. It spiked to really high concentrations in their blood, and I'm talking up around 20 millimolar. And then the problem was, at the 90 and 120-minute mark, it didn't come back down. And these are the classical early signs of mature age diabetes, this sustained elevated glucose concentration in their blood after consuming carbohydrate. So I kid you not, for these 2 individuals here, they were just normal old students, came into the biochem lab and walked away with a suspect mature age diabetes diagnosis. And in both cases, they went away and repeated this test in the clinic and confirmed exactly that. So this is a classical test for mature age diabetes. After feeding a bolus of glucose, the concentrations in the blood spike, and then over a period of time return back to normal levels. And this occurs largely through the action of insulin. But in a mature age diabetic patient, after consuming a bolus of glucose, their concentrations spike in their blood, but are maintained at a high level for a sustained period of time afterwards. And this is dangerous to your health. So therefore, maintaining glucose homeostasis within concentrations ranging between 4 to 5 millimolar is absolutely crucial. And this is achieved through 4 main mechanisms. The first is a bit of a no-brainer. It just comes back to the amount of carbohydrate you eat, in effect, defining substrate availability. The second regulator of glucose homeostasis are the actions of hormones, in particular insulin, glucagon, and adrenaline. The third is the role of different tissues within your body, in particular your liver and your muscle and your fat tissue, as these are the major storage depots for glucose. And then lastly, the fourth mechanism is via the regulation of enzymatic pathways. In particular, these are the pathways that utilise and store glucose or mobilise it when it is in limited supply. So let's start with the really simple ones, including carbohydrate intake, and then the channeling of that glucose to the tissues that really need it. Now, the nice thing about being a monogastric, as a human, we can absorb glucose directly from our gut. And for example, I've already talked about how when we consume starch, we release sucrase in our small intestine, enabling us to effectively digest and absorb that sugar. And this gives us a direct supply from our gut into our bloodstream. Ruminants, on the other hand, don't directly absorb glucose from their gut. Instead, when they eat carbohydrate, it ferments inside their rumen. I've already described the value of this symbiosis, with the microbes getting access to the sugar in cellulose, fermenting it, and excreting volatile fatty acids, in particular butyrate, acetate, and propionate. Now these are absorbed, and particularly the acetate and the butyrate are used for energy or stored as fat. However, propionate is specifically targeted towards glucose production, as it, as a substrate, is gluconeogenic. That is, you can turn it back into glucose. And this is exactly where a ruminant animal gets a large portion of its glucose supply, derived from propionate in its diet. Now, do you recall how I pointed out that ruminants operate at a lower homeostatic concentration of glucose? Well, the reason for this is that they absorb less from their gut. They have to make their own, and as a result, these animals have adapted over the eons to operate in the presence of less circulating blood glucose concentration. Therefore, at homeostatic levels, it's completely normal to see circulating blood glucose concentrations of 3 to 4 millimolar in ruminant animals. Now, the next step is to transport that glucose from the blood into your cells. And this is done via facilitated passive diffusion, facilitated by your GLUT transporters. Now, these are transport proteins that exist on your cell membranes, and they are excellent at transferring glucose into your cells. But this really starts to get interesting when you look at the kinetic properties of these different GLUT proteins. I'll show you this on the next slide. The table you can see here shows 4 of the main GLUT transporters: GLUT1, GLUT2, GLUT3, and GLUT4. These GLUT transporters are expressed at differing levels in differing tissues, and you can see that listed in this column here. Now, as I said, the kinetic properties of these transporters differ, and you can see that in the column listing their Km values. Have a look at GLUT1 and GLUT3, for example. They've got really low Km values. By contrast, GLUT2, this one here, has an extraordinarily high Km value. And GLUT4, this one at the bottom, has a Km value that really matches the homeostatic concentrations of glucose quite well. The other intriguing thing about GLUT4 is that it's also responsive to insulin. What this means is that when insulin is circulating and binds to its receptors, it will cause GLUT4 to migrate from inside the cell out to the cell membranes, where it will start to actively transport glucose into the cell. Now, there's a certain logic to all of this, based upon where these GLUT transporters are expressed, that helps to channel glucose to those tissues that need it most under differing metabolic states. It's closely linked to their Km values, which I'll explain in the next slides. Now, first of all, let's just recap what this Km value actually means. The activity of any enzyme is dependent upon the concentration of substrate that it's got access to. Now, here we're talking about GLUT transporters, so therefore its substrate is plasma glucose. Km refers to the substrate concentration that gives you half of the maximal rate of that enzyme. Now, if you recall back to that table on the previous slide, I described GLUT1 and 3 as having a really low Km value, down around 1 millimolar, which you can see here. What this means is that even at really low glucose concentrations, GLUT1 and 3 can still operate pretty well. And in fact, they'll hit their Vmax up around 2 to 3 millimolars. That's as fast as they can go. They're completely happy. Now, crucially, can you remember where you find GLUT1 and GLUT3 transporters? Yep, they're in your brain and in your central nervous system. So what this means is that even when blood glucose concentrations are really slow, these GLUT transporters are still doing a pretty good job of finding glucose and transporting it into the cells of your brain. Now that's really important because your brain is completely dependent upon glucose as an energy source. And if your brain stops working, well then you're in real strife. Now the next one along, this white line here, represents GLUT4. And you'll remember on that previous table, we said that the Km value for GLUT4 was sitting at about 5 millimolar. Now you find GLUT4 in a range of places, but crucially, you find it in your muscle and in your fat tissue. And the other really vital thing about GLUT4 is that it's insulin responsive. So imagine what would happen if you were well fed. You'd have high levels of circulating glucose, so therefore the concentrations would be elevating up here. You would also have a corresponding spike in insulin concentration. And GLUT4 was an insulin-responsive GLUT transporter. So all of this means that when you're well fed, your body is able to channel a whole stack of that glucose back into your muscle and into your adipose tissue for storage, either as glycogen or as fat. Conversely, if glucose concentrations start to decline because you're starving, you'll have less circulating insulin, so therefore less activation of GLUT4 transporters. And with these lower glucose concentrations, you'll start to see the activity of that transporter diminish quite heavily. So that means when you're starving, you'll stop absorbing glucose into your muscle and your fat tissue. And that's a pretty good outcome. If you're starving, you want to spare glucose for your brain. You don't want to be storing it in muscle or in your adipose tissue. Now the last one here is GLUT2, which is this green line here. And you can see it's got a really high Km value, up around 12 millimolar. This means it's got a relatively low affinity for glucose. Now the other thing not to forget is that enzymes that have got a really high Km value also tend to have a really high Vmax value. It's a bit like having a V8 engine that's only got fourth gear. So if you're trundling along through the suburbs with a pretty slow pace or really low glucose concentrations, this particular enzyme is really going to struggle. But the second you get it up to high glucose concentrations, or if you like, out onto the freeway and stuck in fourth gear, you really start to fly. So this means that any tissue equipped with GLUT2 transporters is going to be able to transport truckloads of glucose into its cells when you're in a well-fed state and you've got really high concentrations of glucose. Whereas the second you start to starve and your glucose concentrations fall away, that tissue will stop absorbing glucose. And this is exactly what happens in the liver. One of the liver's most crucial roles is to act as a glucose sponge for your gut. When you have a high carbohydrate meal and you've got sugar flooding into your bloodstream, it drains from your gut into the mesenteric veins, which feed into the portal vein. And that plugs straight in to your liver. So your liver, which is equipped with these GLUT2 transporters, can sit there soaking up that massive spike in glucose, which follows a high carbohydrate meal. In effect, this protects your peripheral blood flow from those huge spikes in glucose that they would otherwise see. So your liver, with its GLUT2 transporters, is acting as like a frontline defense against these massive spikes in glucose and hyperglycemia. So let's walk through this logic one more time using the following series of three graphs. First of all, consider a circulating glucose concentration of 16 millimolar. So this would be following a high carbohydrate meal where you've got really high circulating levels of glucose in your blood. The GLUT1 and 3 transporters, which you can see in the top graph here, and these are the ones that are expressed in your brain, they'll be perfectly happy, but they'll be operating at Vmax. In effect, they have no capacity to respond to changes in glucose concentration up at this level. They can neither speed up nor slow down. They're just sitting at Vmax. And you call zero order kinetics. Similarly, the GLUT4 transporters here, which of course you'll find in your muscle and in your adipose tissue, they will also be sitting close to their Vmax. Therefore, once again, they've got relatively little ability to speed up if glucose concentrations go any higher, and therefore they're also sitting at zero order kinetics. But our GLUT2 transporters, which are expressed in our liver, are most certainly not sitting at their Vmax at 16 millimolars of blood glucose concentration. So in effect, if glucose concentrations were to go higher still, they'd be able to speed up further, and hence we call these first order kinetics. So in effect, in the well-fed state, the brain and the central nervous system, which expresses GLUT1 and 3 transporters, will be absorbing all of the glucose that it requires. The muscle and the adipose tissue, which express these GLUT4 transporters, will also be absorbing huge amounts of glucose. And the liver, which expresses these high km GLUT2 transporters, will also be absorbing huge amounts of glucose, in effect acting as a type of sponge, protecting your systemic blood supply from any further elevation in glucose concentrations. Now let's look at the next scenario. It's several hours after your last meal and your plasma glucose concentrations have fallen back down to homeostatic levels. So we're talking back at 5 millimolar plasma glucose concentrations. Now in this scenario, your brain and central nervous system expressing the GLUT1 and 3 transporters will still be sitting at Vmax, at zero order kinetics, and in effect absorbing all the glucose it can possibly need. Alternatively, your muscle and your adipose tissue, which expresses the GLUT4 transporter, will be absorbing significantly less glucose as the concentrations have fallen and it's come well off Vmax. But also important is that the insulin levels circulating in the blood will have diminished as well. And of course, insulin is also crucial for the activity of these GLUT4 transporters. Therefore, at homeostatic glucose concentrations of around about 5 millimolar, relatively little glucose will be getting absorbed into the muscle and the adipose tissue. And lastly, the GLUT2 transporters, which are expressed in the liver, will have come right off in their rate of activity. That means that relatively little glucose will be getting absorbed into the liver. And this is precisely what you see under homeostatic conditions, with the liver absorbing relatively little glucose and in fact taking a greater role in actually releasing glucose back out into the bloodstream to help maintain glucose homeostasis. Now last of all, let's get down to starvation levels of glucose. So here I'm talking down at around about 2 millimolar plasma glucose concentration. Now you remember that I pointed out that if you get down to glucose levels this low, it can be pretty bad news because you'll start feeling their effects. Well, to begin with, have a look at the brain, which expresses these GLUT1 and 3 transporters. Down at 2 millimolar, it's still chugging along pretty close to its Vmax. So at starvation levels, the brain is still accessing a good supply of glucose, which is quite crucial for you to stay alive. However, if you go to concentrations below this, you'll suddenly start to see a decline in glucose transport into your brain. And you'll feel the effects of this pretty quickly. Brain function starts to diminish, And for want of any other way of saying it, you start feeling pretty doughy. Turning next to the muscle and the adipose tissue expressing GLUT4 transporters, down at those low concentrations, you can see that the activity of GLUT4s are really backed off. But in addition to that, during starvation, there will be very little insulin circulating. And as such, you'd expect to see very little GLUT4 transporter sitting on the cell surface, actively transporting glucose into those tissues. And of course, for the liver, where the GLUT2 transporters are expressed, at these really low blood glucose concentrations, you'll see hardly any activity of that glucose transport. And in a heavy-duty starvation scenario, the liver will be actively producing glucose and releasing it back out into the bloodstream. And the low affinity of this GLUT transporter effectively stops the liver from reabsorbing that glucose back inside its cells. So you can see that simply by controlling which of these glucose transporters are expressed, and through the variation in their Km, your body has got excellent control over the delivery of that glucose to those tissues that really need it under the different metabolic scenarios. And this is a great example in biochemistry where such a simple form of regulation can exert such massive control over in this case, the partitioning of glucose into different tissues. Now, the following 3 slides simply recap this glucose partitioning story. Firstly, there are the low Km and high affinity GLUT1 and 3 transporters, and these are expressed in glucose-dependent tissue, such as your brain and central nervous system, and even your red blood cells, ensuring a constant supply of glucose to those tissues. Secondly, we've got the low affinity and high Km GLUT2 transporters, which are expressed in your liver. And these are relatively inactive during starvation, but highly active during the well-fed state when you've got big spikes in glucose concentration in your blood. These effectively enable your liver to act as a sponge, protecting the rest of your body against extremely high spikes in blood glucose. And lastly, there are the GLUT4 transporters expressed on your muscle and in your adipose tissue. These are insulin responsive, so generally only active in the well-fed state, and they help to partition excess glucose into your muscle and your fat tissue when you're well-fed, enabling you to store that as glycogen or as fat. There is one other story that I'd like to tell you about, as it ties in nicely with the GLUT transporters. It's to do with the release of insulin from your pancreas. Now, your pancreas is located in your gut, and its tissues interface with the mesenteric veins that are draining your gut. At this tissue interface, your pancreas actually expresses GLUT2 transporters. Now, of course, these have a low affinity and a high Km for glucose, so they don't actually start transporting until there's a big spike in glucose concentration coming from your gut, which is exactly what you would see after you've had a high carbohydrate meal. Now, when your pancreas starts to absorb glucose into its cells, its response is to release insulin back out into the bloodstream. And of course, this insulin can then circulate around your body, activating the GLUT4 transporters on your muscle and your adipose tissue, getting them ready to absorb glucose as it drains from your gut and enters your peripheral blood flow. In some ways, this is like almost an early warning system for your body. It's detecting the glucose before it even gets to your liver, and it's activating that big tissue depot of your muscle and your adipose tissue, getting it ready to soak up that extra glucose that's suddenly available. So that's just fantastic. It's underpinned by something as simple as the Km of the GLUT2 transporters. So to finish with, I thought I'd just re-emphasise some of the main glucose-dependent tissues in your body. Your erythrocytes and also your brain and your central nervous tissue are all glucose-dependent tissues. As such, they're equipped with a GLUT transporter, GLUT1 and 3, that ensures a constant supply of glucose to those tissues, even when you're starving. Muscle tissue, on the other hand, is generally not exclusively glucose dependent. However, type 2 fast glycolytic or white muscle is more glucose dependent, but this generally will have adequate access to glucose via its GLUT4 transporters. And lastly, the liver is, well, it's completely not glucose dependent. It acts as a glucose sink when concentrations are really high, but the rest of the time, and particularly when you're starving, its role is to do the opposite and release glucose out into the bloodstream. Therefore, it's equipped with the low affinity GLUT2 transporter. This can accelerate massively under high glucose concentrations, but when you're starving and the levels are low, it will transport and therefore enable the liver to absorb relatively little glucose from the blood. So in summary, we've recapped carbohydrate structure just as a starting point to get everyone's heads around the basic structures of carbohydrates, particularly monosaccharides, disaccharides and polysaccharides, and also how they're bound together. We've looked at the tissue dependency for glucose and particularly discovered that the erythrocytes in your brain and central nervous system have an absolute requirement for glucose, whilst your liver, for example, has very little requirement. We've also looked at glucose homeostasis and how the regulation of glucose is absolutely crucial in your bloodstream. And we've looked at how your body uses GLUT transporters to partition glucose into different tissues under different metabolic states, all quite simplistically linked back to the Km of those GLUT transporters. And just to finish with, I've got another series of questions that will help you recap the material that we've covered in this lecture. You should certainly go through these questions and, using the information presented in this lecture, feel confident that you can answer each of them.